Okay, good afternoon, everybody. This is Nelly Poyak from Dana Farber, and this is week 2 of our Connect Science seminar series. Thank you, everyone, for joining us today. And as a reminder, the talks will be uploaded at the link that's shown on this first slide. And at the end of the talk, we will have questions. There is a Q&A on the Zoom screen that you can submit questions. Some of them we will be able to answer live with the speaker. The rest will forward to the speaker and he will be able to answer it later on after the seminar. So it's my pleasure to introduce today's speaker, Joan Massaghi from Memorial Sloan Kettering Cancer Center in New York. Joan is the director of the Sloan Kettering Institute and a member of the Cancer Biology and Genetics program, and also professor at the Weill Cornell Graduate School of Medical Sciences. Joan is an internationally recognized leader in of the cancer metastasis and the TGF-beta signaling field. He published many of the seminal papers in this area. Today, his title of his talk today is Understanding Metastasis. So thank you, Joan, for joining us today.
Well, thank you, Nelly, and co-organizers for having me included in this session in these very special times. It's a pleasure and an honor to share the work of my lab with the entire community that is listening in. So today I would like to tell you about our efforts to understand the metastatic process, its operating logic, and to take us to a future where we will progressively no longer regard this problem as the problem that it is today, still the cause of 90% of deaths from cancer. More than becoming fixated in molecules or pathways or stromal components of the microenvironment, which of course are very important, I would really like to try and take us down an understanding of what the process is and what are the cells in whose hands it is to reinitiate and reproduce the tumor at distant sites. Metastasis is the name given to two things. One, a process, the process that starts with the development of a malignant tumor somewhere. That includes cells that have high motility, and the ability of these cells to then infiltrate and overtake distant sites. Metastasis also refers to the end event, the end product of this whole process, the emergence of disseminated disease, which can be treated and is treated tremendously. However, it's very difficult to eliminate, to eradicate, and to cure. And it's many of us who are in a quest to change that. What we know about metastasis? Well, we know a lot about it. We know that the dissemination of cancer cells throughout the body, through the bloodstream mainly, but not exclusively, lymphatics, nerves, and other devices, and other routes, that dissemination occurs early. However, overt metastasis occurs late, months, decades even, after the initial emergence of a tumor and its diagnosis a few months or a couple of years after it got initiated. Most disseminated cells, most cells that leave the tumor die. The ones that survive almost uniformly become dormant, become latent. Metastasis doesn't develop in most cases alongside the tumor within a few weeks of the tumor developing and having started to disseminate cells. It takes a long time, and these cells have survived in a dormant state. The colonization of the organs that they have infiltrated requires special traits. For one, it requires the ability of these cells to be able to traverse the endothelial barrier, the vascular barrier that entraps them if they are coming, as most of them do, through the bloodstream. They also require the ability to interact profitably with the new host environment. In many cases, through many examples that we and others have discovered and described, the host environment, its immune components and other components are not dealing graciously with intruders. They eliminate, they kill the cancer cells that manage to extravasate. These cells have to develop traits to confront this environment, as well as to eventually overtake it, co-opt it to their benefit. We know a fair number of metastatic traits, molecules, pathways, processes that are involved, engaging both the cancer cell as well as specific components of the stroma, cells, as well as matrices and other components, metabolites. In spite of a very extraneous search for a genetic origin of that, like the rest of the steps in tumorigenesis that have preceded metastases, have been revealed to have a very extensive genetic basis, genetic mutations. For metastases, we and others, especially others who have devoted even more time than we have, through sequencing, to identify metastases, specific genes that mediate the specific functions of the metastatic process, metastatic colonization process, have come out nearly empty-handed. The basis for the traits that give rise to metastases has to be found in changes in the ability of cells, cancer cells in this case, to resort to the normal processes that they have, to use them in this selection process and cascade that is metastasis. However, these functions, we think, and we certainly hope, can be targeted for therapeutic advantage. What is that we would like to know that I will be emphasizing with some examples from recent work in the lab in this talk? Well, we would like to know what is the origin and nature of the cells that have the capacity to initiate metastasis. We expect they are stem cells of some kind. Are they the same stem cells that maintain tissue homeostasis in the tumors, in the tissues where the tumors originated? Are they cells that are involved in normal regenerative processes? Are embryonic state cells? What are they? How is it that they enter dormancy, and that dormancy protects the cells from clearance by the immune systems and perhaps other defenses? What is that these cells need in order to resume tumor growth? What do they need to decide to re-enter the cell cycle? And once they do that, how are they going to cope with that environment from which they have self-protected by entering quiescence? Finally, which of these would provide druggable targets for the elimination of metastasis-initiating cells? Note in the cartoon that there is an important U-turn arrow. This arrow means that once metastasis emerges through an outbreak of these latent cells, these may well be treated: radiation, chemotherapy, immunotherapy, combinations thereof, with extensive, in some cases, elimination of the metastatic mass. But only to see that a residue, very minor residue, is able to quickly restore metastasis. We believe, without proof, but with a strong impression about it, that the resources, that the devices that these cells resort to after treatment in order to resume metastasis are probably the same ones that they use to seed metastases months or years later in the first place. So what we are trying to find applies to both, hopefully, the seeding, de novo seeding of metastases, and the relapse of metastatic lesions that have responded well to treatment. How did we get into this? Well, Nelly Poyak, in her introduction, has already made reference to our long-standing interest on TGF-beta signaling and mode of action, the highly contextual nature of the response of cells to the cytokine, and how this contextual nature becomes dramatically apparent when the context is cancer. TGF-beta is a well-known tumor suppressive signal. It induces cytostasis in normal cells, induces apoptosis in pre-malignant cells. The pathway is inactivated genetically in a number of malignancies, gastrointestinal cancers, especially colorectal, and most frequently pancreatic cancer. However, when cancer cells, carcinoma cells at least, solve this problem, the problem of being suppressed, being induced to apoptose in response to TGF-beta, then they are very happy to use the factor and what remains of their pathway for metastatic dissemination, seeding, and colonization. That was a striking observation in the late 90s when my lab and others had labored to understand how TGF-beta acts by having purified the factor, identifying the receptors, membrane receptors that mediate TGF-beta signaling downstream to transcription factors called SMADs binding to promoter regulatory regions of target genes, including among these genes, cyclin-dependent kinase inhibitors, one of which is P27-KIP1, and there are others. And the expression and mobilization of these inhibitors clasps on the cyclin CDK complexes, cyclin-dependent kinases, to induce cell cycle arrest. In more recent work, Chuck David and others in the lab have answered the question, and how is it that TGF-beta induces apoptosis for tumor suppression in premalignant cells that contain KRAS mutation? Again, when these cells bypass these tumor suppressive actions, they remain competent to use TGF-beta now to their own advantage, as first exemplified by a collaborative study that we did with Teresa Geis and her colleagues in the late 90s, showing how in triple-negative breast cancer metastases to the bones, highly osteolytic metastases, TGF-beta is a strong supporter in a vicious cycle represented on the slide, in which TGF-beta release from osteolysis, destruction of bone matrix by osteoclasts, acts on the cancer cells to induce the further expression and production of mediators of osteoclast mobilization. Summarizing these and many other factors that are involved in the TGF-beta promotion of metastases, the scheme shows at the bottom how at the carcinoma stage, cells have one way or another canceled out the tumor suppressive actions of TGF-beta with elimination of the receptors or SMADs, in which case TGF-beta is still helpful if they produce, the cancer cells produce it, or activate what cells in the microenvironment produce, TGF-beta they produce. That TGF-beta will act in support of tumor and metastasis development by acting on cancer-associated fibroblasts. We will see an example of that later in induction of fibrosis and intertumoral fibrosis, and also very importantly, in creating an immunosuppressed tumor environment that favors tumor growth. If the cancer cells at the bottom have eliminated just the tumor suppressive response arm of the pathway, then they get even more. They can use TGF-beta themselves, respond to it with tumorigenic EMT and the expression of organ colonizing genes and functions, such as the ones in the upper panel from the example of osteolytic metastasis. So we wanted, after this lesson, that cancer cells did not need here to resort to any additional mutations beyond those that disrupt tumor suppressors or that activate oncogenes. Once they have that, they can self-select by accessing their genome through appropriate changes or by the acquisition of appropriate cellular stages. They can access a number of functions that support their ability to form aggressive metastatic colonies. To understand this more systematically, we took on to deconstruct the metastatic process by taking the following two complementary approaches. We took cells derived from tumors at two different stages, either from primary tumors, tumors that have not yet generated ovarian metastasis, but have the ability to disseminate cancer cells and seed organs, or from patients who have disseminated aggressively growing metastatic disease. From the first, that we will see a little later, we can try and select, using the mouse as a cell sorter of sorts, cells that have the ability to install themselves as latent disease. Whereas from the latter kinds of cell sources, cell lines, or primary cultures from circulating cells from patients, one can, and we were able to isolate organ-selective metastatic cells. The protocol involves 7 steps. First, use cancer cells from patients with multi-organ metastases for this latter purpose. Isolate cells with selective metastatic tropisms by cycling them through mice. Identify candidate metastasis genes by comparative omics, transcriptomics, or if you want microRNAs, you will get those, or if you want metabolites, you can get those, associated with the ability of these cells to show concrete high metastatic ability in lung, bone, or brain, as in these examples. Filter these candidate genes for association with clinical outcome. Which of these genes associated with metastasis in the mouse are also associated with relapse to that particular site in patients. Functionally validate these metastasis genes as mediators of metastasis. Determine the mechanism of action as well as their value as potential targets by knocking them down and or by using drugs that have been developed or that you can pursue to antagonize their function. This has been successful, this approach, many times over in the lab, leading to many reports that you can consult if you are interested. It's been applied to CNS metastases, to the brain, to the leptomeninges, bone metastases, lung metastases, principally by breast and lung cancers, but also in renal cancers and colorectal cancers. The analysis of the functions of these genes that fulfill all these criteria have given us mediators of extravasation, mediators of seeding, by this meaning those that help these cells cope with the stress of having just entered a distant, strange to them tissue and are dying, are being killed by that environment. And finally, those that are useful later, especially later, those that allow these cells to have survived for a long time in some cases to co-opt the environment. Some of these genes have given us answers to the old question, and how does TGF-beta promote metastasis? The osteoclast activating gene examples from before, and others for lung metastasis. Others have already reached clinical trial stage. CXCL1 and 2 clinical trials being pursued by colleagues here in lung adenocarcinoma, as well as the example that I will show in more detail in a moment, the mediators of brain colonization by the establishment of links between cancer cells and astrocytes. This is the work championed by Qing Chen and Adrian War in the paper that is summarized here with this scheme, but based in prior work from Manuel Valiente, who in the lab discovered that when metastatic cells on the left in green have extravasated, emerged from, in purple, capillaries in the brain of a mouse, the cells having been inoculated into the arterial circulation and allowed to reach all organs in the mouse. This one has emerged and is confronted by astrocytes in red. Astrocytes are there to deal with intruders, to produce, mobilize production of killer cytokines, Fas ligand, and probably others, to cause apoptosis. This cancer cell is competent to produce aggressive metastases in the brain because it has an antidote against that. Manuel showed the production of SERPIN inhibitors of the protease that liberates bioactive Fas ligand, allows the cells to be self-protected from killing. But what we learned, and was known in fact, is that as these lesions, if this cell divides and forms a metastatic lesion in the brain, these cancer cells continue to commingle with astrocytes. We know as cancer biologists that if a tumor has a lot of something, even if that something is supposed to, in principle, protect against tumor growth, that something is probably by now being used for tumor growth. And this is the case again here of astrocytes. Cancer cells, we want to define, generate gut junction proteins that steal astrocytes, induce them to form gut junctions, connexin 43 with the help of protocadherin 7, and they use the gut junctions to transfer to the astrocytes factors that allow the astrocyte or induce the astrocyte to support the cancer cell. The factor that took the lead, or the component that took the lead, was cyclic GAMP, the dicyclic nucleotide product of cGAS in response to the double-stranded DNA that cancer cells with genomic instability have in their cytosol. This factor activates the STING pathway, except the cancer cell has canceled the STING pathway because that would bring the cancer cell trouble, would attract the attention of unwanted attention of NK cells and other mediators of immune attack. Instead, the cancer cell through the gap junctions passes CCAM to the astrocyte, in which cell the pathway, the STING pathway, is completed with the production of interferon alpha and TNF, which in this context provide, and at these levels, provide survival and growth signaling to the cancer cell. There are inhibitors of negative regulators of gap junctions, meclofenamate and tonabacart, that in mouse models and now in patients are showing very promising effects at preventing the outgrowth and blunting the growth that may already exist in brain metastases in patients. Clinical trials in progress by Adrian Boir here at MSK. So many of these genes and studies, genes and factors and mediators of metastasis, the studies that have described their biology have brought us one big leap forward, as done by this lab and many other labs, in our understanding of metastatic colonization. However, progressively we realized that there was something very important that we were leaving behind by obsessively concentrating only in the aggressive outgrowth. How about the cells that are the repositories of these capacities, the metastasis initiating cells? The cells that are depart from the tumor, survive the ordeal, and install themselves in different organs. We know that these cells have tumor regenerating ability because they will do that. They can enter quiescence but come out of it. They are somehow evading immunity, even though they are solitary cells, not within a protective tumor microenvironment. They are exposed to immunity on all four sides, yet they are not being cleared. And they are, of course, chemo-resistant. They are resisting adjuvant chemotherapy that is being given to these patients. So for that, we resorted to the other approach, the approach of isolating metastasis-initiating cells that can seed latent disease. This was the work of Srinivas Maladi, now at UT Southwestern, who patiently took cell lines derived from primary tumors, stage 1 or 2, in breast cancer patients or lung adenocarcinoma patients, tumors that had not yet metastasized overtly, and take these cells and ask whether he would be able to isolate by placing them in the mouse bloodstream, cells that would live for months without taking off and forming metastasis in these mice. And he was able to, and was able to describe, begin to describe in a very rudimentary level at that point in that paper, the nature of these cells. First of all, these cells install themselves as solitary cells. On occasion, one sees clusters. These clusters, regardless of whether the model is from a lung adenocarcinoma or a RAS mutant lung adenocarcinoma or a HER2 mutant breast cancer, these clusters are growing, are proliferating. However, they don't prosper. They get somehow eliminated. If he examined the mice 1 month later and 2 months later, there would be no larger clusters. So somehow, when the cells proliferate, they are unsuccessful at breaking through, with some exceptions, where mice will go on to develop a lethal metastasis, just like it happens in people. When these cells were analyzed, they were found to express signatures of adult stem cells from the tissues they were derived, including markers such as SOX2 and SOX9, which are markers and master or regulatory transcription factors that define a specific progenitor states. For example, on the right bottom, we see that in the lung epithelium developmental process, also recapture parts of it in normal lung regeneration, lung epithelial regeneration. SOX2 and SOX9 play important roles and define two different stages. SOX2, a somewhat more primitive stage than SOX9. There's something else that Srinivas observed, which was a clue to the question of what is that keeps these cells latent. these experiments were being done with human cells in athymic mice. These mice lack B and T cells, but still have macrophages, NK components, components of innate immunity. It is in these mice that one can inject these cells, allow them to settle as metastases, latent metastases, and on occasion, in blue graph, in the graph on the right, on occasion, a mouse will develop a lesion, spontaneous outbreak, as in people. However, when Srinivas eliminated NK cells, depleted NK cells of these mice, there was a massive induction of metastatic growth throughout. And this was the finding that led to the model that is depicted here, which is that as cells with metastasis initiating capacity emerge and install themselves as latent entities, they are not dormant forever, for years. They are apparently trying constantly to re-enter the cell cycle as fluctuations in the signals that maintain them quiescent allow that. And on entering the cell cycle, they will attempt to reproduce the tumor, but they will be stopped on their tracks by innate immunity that will eliminate them. The entry into quiescence, we know, is voluntary, it's proactive. It's not because the environment makes them do that. They themselves known to respond to TGF-beta, we have learned, by first undergoing an EMT and then falling into quiescence. Producing DKK1, an inhibitor of Wnt signaling, thus depriving their microenvironment of Wnt mitogen for their growth. As the balance in these signals changes and a growth occurs, NK cells, and I'm sure other defenses are there to prevent that from happening. So metastatic dormancy is based on a latent state, really, where cancer cells at any point in time, a portion of the population are trying, giving rise to those tiny clusters that are observed in the experimental model, but that are eliminated when you look again. So this is the summary of what we know so far about the metastasis-initiating cells in these models. What else do we know? Well, we know that we have questions, like what is the nature of metastasis-initiating cells? What is their origin? How do they manage to initiate growth when they come out from quiescence? And of course, how could we eliminate them? Reiterating the questions that I posed at the start of the talk. So I'm going to share now some recent work on several of these fronts, not necessarily in this order, from the lab. Some of this work continues on the model of lung adenocarcinomas, or the case of lung adenocarcinomas, and this work performed and published recently by Aisley Loughney in close collaboration between my lab and the lab of Dana Farber here at MSK. Single-cell analysis of samples derived from patients, primary tumor, metastatic samples, as well as normal non-tumor lung tissue, gave rise to the picture TSNE plot that you see on the left, where a majority of the cells in the tumor, as known to pathologists and lung oncologists, are actually not cancer cells. Cancer cells are a minority, roughly 20% or so. Then there are niche cells, mesenchymal progenitors, epithelial cells, etc., and a large number of immune and inflammatory cells. That's the picture. Analyzing these with the questions that I posed a moment ago in mind, I actually wondered whether the collection of normal epithelial cells present in these tumors would in any way resemble, in the stages reflected by the transcriptomes, the normal types of epithelial cells that exist in the lung. Alveolar epithelial cells, type 1, type 2, basal, etc. And how about cell types that are known to emerge during lung regeneration after lung injury, as described by experts in that field? The primary tumor showed to contain among its epithelial carcinoma cells, cells that were a match for each one of these stages, transcriptionally at least. But a majority of the cells in the primary tumor, cancer cells in the primary tumor, were in these gray zone, in these gray arms, which means undefined, mixed up phenotypes. When you look, this is now the epithelial cells, the carcinoma cells from primary tumors. When you look at carcinoma cells from metastasis, the picture was much different. There, almost all cells could be accounted for as corresponding to concrete, known stages of long epithelial development and or regeneration. Primitive stages, embryonic stages, in fact, that lack expression of SOX2 or SOX9, but express SOX17. Very primitive cells. Primitive cells also that are in a quiescent and EMT state, expressing high levels of vimentin and SLOC or SNAIL2, that's markers of EMT, are also devoid of these mediators or determinants of more advanced progenitor states, such as SOX2 and SOX9. So we found that to be remarkable, that how faithfully and clearly a metastatic carcinoma cell population would provide a portrait of the states, transcriptional states at least, that exist in normal development and very abundantly in regeneration. There were other things to be mined out of this data set. For example, the susceptibility to immune surveillance by these various stages is completely different. For example, the quiescent state, that is NK cell resistant, we know from other work, is very high in class 1 MHC molecules, HLA A, B, and C. They are strongly downregulated as the cells are in more advanced progenitor states, SOX2 and SOX9 double positive states, to reemerge in the SOX9 state. ULBP and other ligands at the bottom. These are ligands, cell surface proteins that are expressed in distress cells to act as ligands for the killer cell receptors, NK cells and cytotoxic T lymphocytes. These also show varying patterns of expression, telling us or suggesting to us that the susceptibility to metastatic clearance is going to be quite selective and specific for different stages in the metastatic cell population. In other words, heterogeneity in metastatic cell populations, carcinoma populations, refers also to the individual cells' susceptibility to mediators of immune cells to mediate clearance. This was just a static portrait, but we could make use now of the model devised by Srinivas Maladi to ask what would happen when, with these various stages, as we track a population of metastasis-initiating cells from the latent state, shown in brown, to the state when they begin to make a few incipient micro metastatic colonies, to the state when they become a more heterogeneous, aggressive metastatic colony. And analyzing the cells by using single cell analytics, we were able first to reproduce and find here also the many subtypes, the many subsets of progenitor states described transcriptionally, identified in the human lung adenocarcinoma primaries and especially metastases. Secondly, they travel with time. That is, the latent metastasis cells were more rich in cells that are SOX2 positive as well as double negative. How the incipient metastases are largely in the SOX2 more primitive progenitor state, and the more overt metastasis is enriched in SOX9. Why is that? Is it because this is happening in a spontaneous mouse, spontaneously in a mouse that still has innate immunity, and the different cells are good at different stages? Or might it be that there is constant pruning of some of these populations selectively? And this is what Ashley went on to find, that in the spontaneous tumors that emerge are dominated, the large tumors, the large metastases, by SOX2 positive cells in red, whereas if one looks at tumors that emerge massively in NK-depleted cells, these tumors have, in addition to red labeled SOX9 positive cells, plenty of SOX2 cells, leading through experiments shown in this slide, done in vitro, of exposing these SOX9 or SOX2 cells to activated NK cells, leading to the finding that the SOX2 stage, it's fine, it's aggressively forming metastases or trying to, but these cells are highly susceptible by virtue of the ULBP ligands factors that they express, highly susceptible to clearance by NK cells, whereas the SOX9 cells are much less so. So that is teaching us two lessons about the nature of these cells and how they develop after they are able to engage spontaneously in an outbreak, as they do in people, presumably. There is developmental stage-specific pruning of these populations by immune cells. The metastasis that results, perhaps not surprisingly, but important to show, to demonstrate here, is a result of a balance, a tug of war between cells that are trying to regenerate the tumor and immunity that is fully successfully during latency or only partially successfully, not successfully, that is when there is a metastatic outbreak. What else do we know about these cells? What else do we know about what they do once they try to initiate metastasis? Here's looking at a lung adenocarcinoma cell that has infiltrated the brain of a mouse, has emerged from the capillaries, and is about to go down its path to try and reproduce the tumor. We see in the movie, which is a time-lapse series of C-stack confocal images, how these cells are hugging very intensely the capillaries that they have just emerged from. They are migrating. It is migrating. It's pushing out of the way as it migrates pericytes. It is, we know, in a clear EMT state, and it will eventually stop to set shop, to undergo an MET, a reversion of EMT, and to begin proliferating. That proliferation will give rise to a sheath around the tumor, as seen in day 4, day 7, day 14 sequence in the upper track of images. If this is a model derived from an aggressive metastatic tumor, a tumor line, tumor-derived cell population, one that already knows to defend itself well from immune attack by the microenvironment. If, on the other hand, one is examining the latent models, we see again that on day 7, they are still, the cells that have emerged, still extended on the capillaries, migrating, but ready to fall into deep quiescence, rounding up and staying that way for a long time. So EMT emerges as an important thing to pay attention to. Of course, many have paid tremendous attention to EMT throughout biology of development, of wound healing, and very especially of cancer and metastasis. What EMT is well known to many of you. The key features are listed here. It is something, it's a normal process that is manifest also in cancer. And when it's manifest in cancer cells, it endows these cells with the ability to migrate, to detach from each other, to infiltrate. And it's also observed associated with states of early progenitor stem-like states. What induces EMT? Well, TGF-beta, not our work, the work of many others, is known to be one of the most prominent, frequent, and potent signals from the environment telling endothelial cells, epithelial cells to undergo EMT. For a long time, it has not been known, however, how TGF-beta triggers an EMT. And also, how these EMTs are coupled with the other programs that the cells may harbor. In wound healing and in cancer, EMTs are coupled with the induction of or the appearance of a fibrotic response, production of massive extracellular matrix, ostensibly for the support of wound healing, but leading to complications in the form of fibrotic disease and including intertumoral fibrosis. However, another instance where a primordial EMT is critical is during gastrulation. There is no fibrosis. What we have there is EMT happening in cells that at the same time, they are differentiating into mesendoderm. So the question is, how is EMT coupled to these other programs? In our metastatic cells, how would it be coupled to fibrosis if this is what's going on, or deep quiescence if this is what's happening in epithelial, latent cells? Addressing these questions is recent work by Jessie Su, in which she leveraged the observation that it's only certain responses to TGF-beta that depend on or are modified profoundly in premalignant cells that have acquired mutations in RAS. Among these responses is the induction of an EMT. Normal epithelial cells, with exceptions, do not produce a strong EMT response to TGF-beta. Mammary progenitors do, but many others do not. Now, if these cells receive or harbor a mutant RAS or any other form of hyperactivating MAP kinase pathway signaling, a number of TGF-beta gene responses become very augmented. We are looking here on the left to SNAIL. SNAIL is the transcription factor that orchestrates, one of them that orchestrates EMTs. The SNAIL response to TGF-beta is very weak when RAS is not, oncogenic RAS is not expressed, but strong induced, along with a panel of other responses seen in the middle panel that are off the diagonal in the presence of strong MAP kinase activating oncogenic RAS signals. Common to this subset of TGF-beta target genes was the presence in their regulatory regions, right next to where SMADs bind in chromatin IP, the presence of sites for a fairly obscure factor, REB1. REB1 also emerges independently. There's a study when doing a hairpin screen for factors, transcription factors, required for the induction of EMT by TGF-beta. So now from two different orthogonal approaches, association of its sites with the genes that are selectively hyperinduced along with the SNAIL by TGF-beta in the presence of RAS, and also as a gene required for these responses, REB1 emerges as the candidate factor that allows cells to respond to TGF-beta with a strong induction of an EMT. What is REB? REB stands for RAS response element binding protein 1. In spite of this name, very little known, very limited studies on it, only a few dozen compared with the other major known transcriptional targets of RAS. Some implication in cancer, known to be important itself in cell lines, mammalian cell lines, or its fly homolog hindsight in mouse in fly genetic studies, important for epithelialization processes. Well, here Jessie was able to show that in the canonical TGF-beta pathway, REB plays a crucial role in endowing the pathway and enabling the cell to mount an EMT. REB activated by RAS-MAP kinase binds to this subset of TGF-beta target genes to their regulatory regions before the TGF-beta signaling. So it is pre-bound, as seen in the upper tracks of the chip tracks in the upper left panel. It's pre-bound in gray, awaiting arrival of receptor-activated SMAD transcription factors, helping recruit these factors and becoming essential for the transcriptional activation of the target genes, SNAIL and others included, but not required at all for target genes represented here by SMAD7, TGF-beta target genes that are independent of RAS signaling. If Reb1 is knocked out, SMAD-dependent induction of EMT, as shown in this example of pancreatic carcinoma, premalignant carcinoma cells, that EMT in independent knockouts is prevented completely. And this is an effect observed in all cell types, RAS mutant or even normal mammary progenitor cells that are known to undergo a MAP kinase dependent EMT. All of that is blocked when Reb1 is knocked out. In lung adenocarcinoma models, for example, for which an EMT is a clear pro-tumorigenic and pro-metastatic function, knocking out of Reb1 prevents EMT, and with that prevents tumor emergence to a shocking degree, to more than we frankly expected from preventing the migration and infiltration of these cancer cells. I mean, in subcutaneous tumors, the cells don't have to migrate very far, they are just growing there, and that was severely blunted. What else may be going on that is downstream of Reb besides the EMT proper? Well, we observe, Jesse did, that these tumors, in this case metastases to the lung, by these lung adenocarcinoma cells, is very abundantly populated by mesenchymal cells or cancer-associated fibroblasts with markers of myofibroblasts, strong deposition of collagen, collagen fibers being stained here in large tumors as well as in tiny micrometastatic lesions. That is already apparent in the second column. Whereas in size-matched lesions formed by the Reb knockout cells, they are growing, they are not going to grow very well, very far. They are completely devoid of these intratumoral fibrosis and the cells, myofibroblasts and CAFs that give rise to it. So that made us realize what is summarized in this slide, which is that TGF-beta signaling together with RAS generates a complex Reb1 SMAD that on the one hand activates an EMT and on the other hand activates a program, a set of genes that constitute a fibrogenic program. Genes that the cell undergoing EMT is expressing to produce signals, diffusible cytokines, that will act in the myofibroblast progenitors in the environment, together with TGF-beta itself also acting directly on them to promote rapid differentiation, expansion, massive accumulation of matrix. Mirroring this in other settings, in development and gastrulation, the principle is the same, except that the program that accompanies the EMT transcription factor activation by Rep-SMAD, these transcription factors for EMT are snail, twist, and ZEB1 has to also play a role. Here, the program that accompanies is EOMS, guscoid, MIXL1, brachyury, and activation of other genes that constitute the series of transcription factors that drive mesendoderm differentiation. So the principle that emerges is that TGF-beta signaling in cooperation with RAS orchestrates EMTs that are different in flavor in accompanying factor depending on the context. But back to our center of interest, the EMT that happens in cells that emerge from vessels and are attempting to initiate metastases, secreting themselves into quiescence in latent models, or going forward in models that are aggressive. If they go forward, they are observed, these cells, forming these sheaths around the vessels, and they will form the tumors, classical massive tumors later, but they start as a tight sheath around these vessels, as shown here. We are looking at the result of 3 cell divisions, 8 cells after one of these EMT undergoing cells has stopped, has arrested, is presumably undergoing MET. These cells are now in contact with each other, are dividing, they are not letting go of the vessel, they are forming this sheath. How do they do that? That is a remarkable function also of these metastasis initiating cells. We want to know more about this function because it may give us clues as to the origin of these cells. What are these cells that arrive and extravasate in distant organs capable of performing this most remarkable beat of cell biology. One of the experiments that revealed this, Manuel Valiente was able to identify the cell adhesion molecule that allows the cells to spread themselves so tightly as they rest on capillaries. This is L1-CAM, L1 cell adhesion molecule. If this molecule is knocked down in these highly aggressive metastatic cells, the cells will still be able to circulate normally, extravasate, stay by capillaries, but in the short hairpin knockdown against L1-CAM, these cells are unable to hug and spread in the capillaries. And with that, they lose also the ability to form colonies by 10 to 100-fold decrease. This is seen here with lung and breast adenocarcinoma cells metastatic to the brain, but has since been confirmed also by EMBRA-ER in the cited paper at the bottom to happen in breast and lung metastasis to bone, to liver, to lung as well as metastases by other tumor types. So L1-CAM is necessary at this moment for reinitiation of tumor growth in different tumor types and in different organ sites. The first general mediator of metastasis that we run into, the first one that is non-tissue or organ specific, but required in multiple organs by multiple sites. Why do these cells need it? What does it do for these cells, L1-CAM? This is the work by EMBRA-ER, now at the University of Illinois, in which he went on to show that L1-CAM cooperates with beta-1 integrins to engage ankyrin, and through integrin link kinase and PAC activate YAP and MRTF transcription factors, major transcription factors of mechanotransduction, to give these cells the green light to proliferate. These cells are exposed to nutrients, oxygen, growth factors. They are cancerous, but they will not grow unless they get the go-ahead from this signal. He's looking at examples of these cells in human metastases, samples from patients in which perivascular breast cancer cells are seen expressing L1-CAM and active YAP. There's a lot that was known about L1-CAM and is present in tumors. L1-CAM is a cell adhesion molecule, and the field was struck in the 90s and since the 90s by how this L1-CAM expressing molecule, the expression being thought to be restricted only to neural development and in the brain, to be appearing over and over in all kinds of tumors. And the higher the proportion of L1-CAM that tumors express, many reports showing, as reviewed in the cited review, showing a poorer prognosis. Well, being a cell adhesion molecule, it must be doing something to support migration, metastasis. Exactly what wasn't clear. Now we know what I just described. But it led to the question of, why do these cells come up with L1-CAM expression, these metastatic cells? Are they ever supposed to have that? And this, to end the presentation, is the work that was recently reported by Karuna Ganesh, now an independent investigator at Memorial Sloan Kettering. Karuna, a GI oncologist, decided to resort to colorectal cancer as the context and model to ask the question, how are L1-CAM cells emerging, expressing cells, positive cells, emerging in a primary tumor that then must prepare them to do what they have to do once they reach a distant site? She was able to verify what others had already described, which is L1-CAM is not expressed in the normal colonic epithelium at all. At the bottom of the crypts where reside cells, stem cells, adult homeostatic stem cells, LGR5 positive, well known by many, there is no L1-CAM positive cell to be found there. However, as tumors emerge and in the invasion front of tumors, there are minorities of cells expressing L1-CAM. The proportion of L1-CAM expressing cells is even higher in liver metastasis and becomes even higher, more enriched for, once there is a response or treatment with neoadjuvant therapy. Is L1CAM important for something? Well, Karuna asked this question by asking whether L1CAM would be important for the ability of these cells to regenerate their tissue. Organoid growth assays are a form of tissue regeneration from cells that have been, in this case, dislodged completely. If they are dislodged completely from normal tissue or from primary or metastatic tumors, these cells are sorted then for L1CAM high or L1CAM low to find that it's the L1CAM high, the subset of cells that have the highest organoid forming capacity. The limited proportion of L1CAM low cells that do form organoids, when examined, that is because they have recovered or somehow express L1CAM them too. These cells are also capable of transmitting the tumor, transplanting the tumor into a recipient mouse, whereas the L1CAM negative or low cells are not. Single cell analysis was used instead of antibodies that do not exist for flow sorting for LGR5 cells in order to answer the question whether L1CAM cells overlap with LGR5. And as the data shown here illustrates, many L1CAM positive cells are low in LGR5. Only a portion of them in orange overlap with an LGR5 high state. We can knock out L1CAM, and these cells will fail completely to form organoids. So L1CAM is expressed by dissociated cells to form organoids and is required by these cells to form the organoids. Interestingly, L1CAM is not expressed in crypts, isolated as such, as intact crypts, and is probably not required by intact crypts to form organoids when these organoids are not formed through an intermediate step of dissociating the epithelial cells, the way that cells need to be dissociated in a metastatic process to circulate and disseminate. Moreover, Karuna went on to find that these strongly L1CAM expressing cells that initiate organoid growth progressively lose the expression of L1CAM. The positive ones remain relegated to the corners of this tumor organoid until and unless you dissociate this organoid to passage it, at which time there is a strong reactivation of L1CAM expression in a portion of the cells that had already become L1CAM low. She was able to work out, Karuna was, the molecular mechanism that keeps L1CAM repressed in the cells by the rest repressor complex that is maintained for as long as there is cell-cell contact between the cells. Dissociation of the cells relieves L1CAM from rest repression, leading to the expression of L1CAM that the cells need to form growths in matrigel, organoid growths in matrigel. I remind you that matrigel is basement membrane, laminin rich, and laminin is the ligand of L1CAM. So that began to remind us more and more of regenerative processes, dissociation of cells, leading them to express a molecule for adhesion that they need for growth. It was time to begin to look at normal epithelium. I already told you that L1CAM is not expressed in normal epithelium. But as it was discovered here by Karuna, when a colonic or intestinal epithelium in the mouse is subject to colitis, to ulceration by administration of dextran sodium sulfate, there is immediate denudation of this epithelium. And within a few days, when repair and regeneration eventually occur, now one sees the massive emergence of L1CAM expressing cells. And these repair layer of epithelial cells stays L1CAM positive for a while. If in this context one knocks out using a conditional allele L1CAM, the result is shown here. These epithelial cells that never express L1CAM or needed it for normal intestinal homeostasis, now they fail to regenerate the epithelium after ulceration, and the mice die of dehydration and diarrhea because the L1CAM expressing cells are unable to reconstitute the normal tissue. Summarizing all this and other data that I have not had time to present, we have the following view, which is that repair and regeneration is in the hands of, as it's increasingly known, specially mobilized progenitor states that emerge from pre-existing stem or progenitor cells through profound plasticity, phenotypic plasticity of these populations, normal populations. When there is severe damage, these populations will emerge through plasticity into phenotypes that express L1CAM, which they require in order to sense basement membrane and to give themselves the go-ahead to proliferate to cover the wound. These replace, it appears, in cancer. Polyp emergence in the colorectal model or in the intestinal cancer model does not involve expression of L1CAM, does not require L1CAM. Polyps are still structurally perfectly fine epithelium. There is just too much of it. But as the carcinoma, invasive carcinoma stage takes place, the tumor progresses to that stage. Now the disruption that this stage creates on itself is accompanied by the appearance of L1CAM expressing cells in the invasion front, the cells that are also passing into the circulation to emerge at the other end, co-opting vessels, using L1CAM once they are ready to start growing, to spread, to activate YAP, to proliferate. Or in the case of the surrogate assay, organoids to do the same there. So we end then with at least some sense of what the answers to the question that I posed at the beginning may be, which is that metastasis is initiated by cells that mimic wound healing processes. They are stem progenitor-like states that can be found if one digs deep in regenerative, developmental and regenerative progenitor processes, endowed with tremendous plasticity, endowed with differences in susceptibility to immune clearance, difference in ability to interact with with their environment for sure. It involves the metastatic process, EMT, driven by TGF-beta, probably other factors as well as immune evasive quiescence, something else that TGF-beta and anything else that impinges on quiescence provides the cells with. Metastatic outbreaks, whose emergence is still a mystery. That is how it is that these cells emerge from quiescence. But when they do, they are going to need certain special functions. We've discovered one in L1CAM. This begins to give some targets of interest, some of them very specific for these moments of early colonization, L1CAM perhaps. Although we believe, as we study this further, that they are used and reused discreetly at all points in invasion from biology, in dissemination, as well as in overt metastasis. So these are the thoughts, progress, and findings that I wanted to leave you with. I want to acknowledge those who have contributed most directly to this work, the current members of the lab, who are now pursuing the work that I presented, recent past members of the lab, including the ones that have just completed these studies recently published, as well as the many collaborators and funders that made all of this possible. And I thank you for your attention.
Thank you, Joan, for this excellent and very comprehensive talk on metastasis. We ran over a little bit of time, so we won't have many questions, but at least one I'd like to ask from, there were many questions submitted online. Many of them relate to the question regarding to these tumor metastasis initiating cells. Are these like related to the cancer stem cells? And if they are these progenitor features, could you make them differentiate?
To the extent that we have studied, in the models that we have studied, they are clearly different from cancer stem cells. Cancer stem cells, where they have been studied, are normal adult stem cells, homeostatic stem cells, in colorectal cancer, for example, that have been the recipient of mutations, that have turned them into stem cells that are unable to differentiate properly, yet they continue to be able to proliferate. These are not the states that are captured in the metastatic initiating a state as we have observed. We thought they would be. There is plasticity. I'm sure they can be interconverting, but the state that initiates metastasis is of states. It's not just one cell or one state. It's a series of states that are in and out of themselves, becoming the other, and so forth. They are not dominated by the classic adult stem cell state that in turn serves as the initiator of tumorigenesis when that state becomes the target of mutations.
Okay. Thank you very much. There are a number of questions related to the immune evasion and immune suppressive mechanism, but maybe you can answer these later on. We will send you all the Q&As later on. So just as with that again, I'd like to thank Joan for the outstanding talk and covering these really complicated and diverse areas. And as a reminder, the talk will be up, the slides will be uploaded on the website so people can later on look at it. And also Joan will have access to the questions that you submitted and he will be able to answer each one of these to the attendees. So thank you again, everyone, for there was a great participation, and thanks, Joan, for the talk. Bye.
Thank you all very much.
Bye-bye.
Bye-bye.